So first of all, I want to say the organizing committee of Cancer Stem Cell to give me this opportunity to, to present my work. So I'll be starting with my presentation. I'll be talking about the expression of CD44 in oral squamous cell carcinoma and metastatic lymph nodes. Oral squamous cell carcinoma, it is prevalent in Southeast Asia, also in certain countries of Southern Europe. I'll just like to brief you about squamous cell carcinoma, especially the oral one. Now there are certain important risk factors, like for example, tobacco consumption. It's very high in Southeast Asia, and that is the prime etiology for oral squamous cell carcinoma. Also, alcohol intake is also one of the important etiological factor for oral squamous cell carcinoma. Now, of late, in certain conditions, HPV, human papilloma virus, has also been the culprit for the oral squamous cell carcinoma. Now, these pictures, they show that oral squamous cell carcinoma can start as a tiny lesion on the tongue, and it can be very, very aggressive. In the third picture, that it is involving almost the entire lower part of the oral cavity, floor of the mouth, jaw, and adjoining structures. These pictures talk about the oral squamous cell carcinoma present in floor of the mouth. The second one is the oral squamous cell carcinoma in palate. So what I mean to say that this is a very, very common cancer, especially in Southeast Asia and also in India, where I belong to. It is the third most common cancer in India. The first one is breast, the second one is lungs, and the third is oral cancer. Now, within the oral cavity, there are certain sites: buccal mucosa, tongue, lower lip, palate, alveolus, gingiva. But out of all these areas, it is most commonly seen in buccal mucosa in almost half of the cases. Now, gender-wise, it is almost twice as common in males compared to the females. Age-wise, it usually involves the people of 50 plus, because what happens, this tobacco chewing or smoking or the alcohol intake, it does not cause cancer very early. It takes around 15 to 20 years. By the time they will be 50, 55 or 60, then they develop this oral squamous cell carcinoma. So it is a very, very, you can say, one of the leading causes of death in India. So a lot of work is going on in oral squamous cell carcinoma, starting from the diagnostic markers, prognostic markers. Various studies are going on. Coming to the CD44, which I have taken as the part of my study, the expression of CD44 in oral squamous cell carcinoma as well as in its metastatic lymph nodes. So a brief introduction about CD44 also. The CD44 is a multistructural and multifunctional glycoprotein. CD44 is a cell membrane molecule that was first identified on lymphocytes and was initially found to have cell adhesion and cell homing functions, also called as Hermes antigen. In 1989, Stamenkovic and colleagues found that a variety of carcinoma cell lines and solid tumors expressed CD44 gene. The CD44 gene has been mapped on chromosome locus 11p13, short arm. The most prolific isoform of CD44 has been termed as CD44 standard, designated as CD44S. The other variant are called as CD44V, V1, V2, V3, and so on. There are multiple variants of CD44. Now this is the structure of CD44. The upper green section shows a constant region of messenger RNA included in every possible variant. The red section is a variant exon that might be selected via alternative splicing. The lower section shows different domains of CD44. Another picture, here we can appreciate these domains much clearly. Extracellular domain, also called as ectodomain, transdomain, transmembrane domain, and intercellular domain. The first 5 exons of CD44 encode an amino terminal globular protein containing motifs that function as ligand binding receptor, mainly for hyaluronic acid. So the hyaluronin is the main ligand for CD44. Now CD44 is present ubiquitously in almost all systems of the body: central nervous system, lungs, epidermis, liver, pancreas, keratinocytes, lymphocytes, macrophages, cervix, bladder, stomach. Also on various cells it has been spotted: leukocytes, fibroblast, epithelial, mesodermal, neuroectodermal, cancer stem cells. Also in the oral tissues, it has been seen on various sites like tongue, gingiva, esophagus, larynx, keratinocytes, stratum basale, stratum spinosum. There are multiple physiological roles of CD44 like cellular adhesion, hyaluronin degradation, lymphocyte activation, lymph node homing, myelopoiesis, lymphopoiesis, angiogenesis, release of cytokines. Epithelial mesenchymal transition. Epithelial mesenchymal transition is a very important topic. Is a tightly regulated and highly conserved cellular process for a cell type changing from an epithelial phenotype to a mesenchymal phenotype. Now, epithelial mesenchymal transition is seen normally in embryogenesis, tissue remodeling, inflammation, tumor proliferation, invasion, drug resistance. Epithelial mesenchymal transition is involved in the acquisition of stemness of epithelial tumor cells, which confers cells with aggressive traits and an invasive phenotype that may result in tumor recurrence and metastasis. CD44 also promotes epithelial mesenchymal transition in many cancer types such as colon cancer, gastric cancer, pancreatic cancer, prostate cancer, liver cancer, glioma, by upregulating mesenchymal markers and downregulating epithelial markers, especially in gliomas by upregulating the mesenchymal markers and downregulating epithelial markers. Cell matrix interaction. CD44 functions as principally dependent on cellular adhesion. This adhesion can lead to interaction between two different cells or between a cell and its pericellular matrix. Important ligands of CD44: hyaluronate, chondroitin, collagen, laminin, fibronectin. The most important ligand is hyaluronate. The proliferating cells express high amounts of CD44. The proliferating cells are typically rich in hyaluronate. Cancer stem cells synthesize hyaluronic acid to attract tumor-associated macrophages. Now, cancer stem cells and tumor-associated macrophages produce oncogenic growth factor, that is platelet-derived growth factor, that maintains the tumor cell in a sustained proliferative phase. So we can deduce that the hyaluronic acid is attracting the macrophages, which in turn are attracting the cells from the matrix, and they are sustaining the proliferation of tumor. Now, stromal cells are known to produce multiple growth factors that regulate stem cell activity and reproductivity. CD44 is used by several cell types to mediate hyaluronate-dependent adhesion. Aggregation of macrophages and lymphocytes is induced by exogenous hyaluronate. Now, recent research has shown another ligand for CD44, that is osteopontin. Now, osteopontin is involved in regulation of inflammation, osteogenesis, angiogenesis. Interaction between CD44 on lymphocyte and a protein known as adressine on endothelial cells of Peyer's patches on the lymph nodes that helps in lymph node homing. So CD44 plays a crucial role in movement and homing of antigen. The CD44 also activates B and T lymphocytes and maturation of stem cell progenitors. CD44 provides proliferation initiating signals from early progenitors of all the three hematopoietic lineages: myelopoiesis, lymphopoiesis, angiogenesis. The proliferation and migration are two features required in angiogenesis are initiated by CD44. CD44 also plays an important role in metastasis. The ability of CD44 to degrade hyaluronate could also be used by tumor cells to assist in its path through the basement membrane and the vessel wall. The tumor cells that metastasize by the way of lymphatic system are thought to imitate lymphocytes entering peripheral lymphatics and traveling to the draining lymph nodes. CD44 assists in distant metastasis by penetration through vessel walls and expansion into foreign tissue. Vascularization of newly embedded tumor may well involve CD44, which has been to be important in angiogenesis. Now, especially in the breast cancer, CD44 and CD24, abnormally they are expressed in breast cancer. The CD44 and CD24 ratio could be a guideline for early detection of neoplastic growth in human breast tissue. CD44 can be used as a therapeutic target. It can be used to abrogate the cancer cell proliferation and metastasis. Like in cancer hepatic cells, when treated with antisense oligonucleotide, showed a downregulation in CD44 expression combined with an increase in cellular apoptosis, which may reduce invasion and metastasis of the cancer. In patients with chronic cell leukemia, targeting CD44 expression in myeloid cells could avoid immature proliferation of white blood cells. It has been demonstrated the CD44 variant 3 functions as co-receptor for fibroblast growth factor and vascular endothelial growth factor. These growth factors are very important in angiogenesis and neoplastic formation. Now, coming to my study, I took up this research project of CD44S, that is the standard variant in squamous cell carcinoma. As per the broader grading, there are three types of oral squamous cell carcinoma: well differentiated, moderately differentiated, and poorly differentiated. So this is an image of well differentiated. We can see that CD44 is present all around the tumor islands, in and around the keratin poles where they present. Very strongly they are expressed. Now this is the moderate variant of oral squamous cell carcinoma. They are present in the islands as well as in the stroma, even in the islands without keratin pearls. In poorly differentiated, we hardly get to see any keratin. We get to see only the tumor cells arranged in the form of strands and cords. They all are overtly expressed by CD44. Now, contrast here is that CD44S is absent in metastatic lymph nodes. I did this research on 30 cases. of various grades of oral squamous cell carcinoma starting from T1 to T3, but I could not find CD44S expression in many of them. Hardly in 1 or 2 cases, it was very weakly expressed. So we cannot comment that metastatic lymph nodes, even if they are fully infiltrated with the tumor cells, they show a presence of CD44 standard. Now these statistics in brief, they depict the same, that CD44S is very well expressed in all the grades of oral squamous cell carcinoma, starting from well to moderate to poor, but it is weakly expressed in metastatic lymph nodes of oral squamous cell carcinoma. Now my future prospect will be to study the variants of CD44 in various grades of oral squamous cell carcinoma and the associated metastatic lymph nodes, and to establish whether any of the variants of CD44 from V1 to V6, whether it can be used as a prognostic marker or not. So in future, I'll be continuing with the various uh, aspects of CD44. My heartfelt thanks to my colleagues in the Department of Oral Maxillofacial Pathology who work with me. Special thanks to Dr. Vidyadevi Chandavarkar, Associate Professor, Dr. Bond Professor. I thank you all for the patient hearing. I hope I have presented it well. If any questions are there, please, I'll be happy.
Thank you very much, Dr. Mishra. I think there is a question from Marina, and she wrote it into the chat. And is it about the expression? Was it higher or lower in normal mucosa than in cancer? This is one question. And the role of CD44 in the interaction with the extracellular matrix, was there any interaction with other signaling pathways? These are the questions that were written in the chat.
Yeah, I would like to answer this. The
Please.
induction of epithelial mesenchymal interaction by CD44, it's not only by itself. There are also interaction with other signaling pathways, like maybe tyrosine kinase pathway or other MEK pathway. It's not only by CD44. Okay. And CD44S expression is, I cannot say it whether it is higher or lower, but it is present in a, you can say, the weak, we call it as a strong expression or weak expression. It is weakly present in normal oral squamous, normal oral mucosa, but in the cancer, it is strongly expressed. That is the difference between the presence of CD44S in normal mucosa and in the cancer. It is much stronger and all over in the and in the tumor cells in the cancer.
Please, yes. Dr. Mishra, may I just clarify? So I studied adenocarcinomas and adenomas of prostate. So I saw that expression of basolateral membranes in non-cancerous glands. It was present in all the cases and rather strong. So do you see any staining in normal squamous epithelium? In normal, I mean non-cancerous epithelium. Squamous versus glandular.
Ma'am, your audio is not clear. I'm not able to interpret.
Sorry, did you see any staining membranous in normal?
You can type in the chat box.
Okay.
Okay. Yeah, it's also very interesting that you didn't obtain any positive cells in the lymph nodes. This is very interesting result. And we are waiting for the question in the chat about the positivity of CD44 in normal squamous cells compared to the cancer.
Done
Yeah. Do you
already?
see the question? So it's about the staining in normal epithelium.
I saw the staining in normal squamous epithelium. Yes, it was a weak staining in the normal squamous epithelium. Yes, it was.
Okay.
It was
Thank
a
you
weak
very
staining.
much.